that's going around, make sure you get a copy of it so you can check your answers. As I said, also make sure that the form that was indicated is the correct form for your exam. So that's one reason why you want to keep that exam test booklet that's got the questions on it. The other reason is because it's a good tool to use to study for the final. I apologize, I got injured in the crush. I don't usually wear paper towel on my thumbs. We were looking last time at enzymes and energy considerations, and so I stopped here where we were looking at how a particular enzyme might work. This is an enzyme called sucrase that breaks down sugars. And by the way, you can always recognize the name of an enzyme because it always has ASE at the end. So sucrase is an enzyme that breaks down sucrose. Sucrose is a disaccharide, and the enzyme up there in step 1 has an active site. That's where the actual molecule of sucrose is going to bind. Once that molecule binds, then you get a shape change in the protein. And what that does is that allows the substrate to fit a little bit better. It also tends to activate the substrate in the sense of changing the bonds or stretching the bonds so that it's more unstable. You're adding that energy of activation that's necessary to get the reaction to go a little bit faster or a little bit more easily. Okay? Water then is added to this molecule to hydrolyze it, and the substrate is converted to its products, its monosaccharides, which are glucose and fructose. Then those products are released from the enzyme, and the enzyme is ready to go again. Okay. Enzymes are unchanged by the reactions that they do. There's only 2 exceptions I can think of where an enzyme is poisoned. One of them is metallothionein. It's an enzyme that actually breaks down or removes heavy metal ions. So if you get exposed to like mercury, then metallothionein sort of sucks up those mercury ions and gets rid of them. The other one is an enzyme that adds sulfur groups in a fairly complicated reaction. But all the other enzymes I can think of are enzymes that are unchanged by the reaction that they do. Okay, so there's a question to ask to see if you remember this stuff about enzymes. Okay, if you were paying attention Friday and Monday, this should be, or Friday and today, this should be fairly obvious. The incorrect statement there is C. Enzymes are used up in chemical reactions. They're not. They're unchanged. Okay, so there's the 2 exceptions, metallothionein and S-adenosylmethionine transferase. You don't need to remember that. Don't worry about it. Okay, just remember they're unchanged by their reaction. I was sort of hoping I'd be able to finish that up on Friday, but I didn't quite get far enough. So we're now on to the next stage of this, which is biological membranes. You remember this chapter has basically 2 different parts to it. One of those parts was energy and energy usage in the cell, and the other part is the part about biological membranes. So we need to take a look at the structure and function of the membranes of cells. It's an important consideration because the membrane is a barrier, right? It defines the cell's inside from whatever is outside, and it's used to maintain different conditions inside the cell than you find outside the cell. How those conditions are maintained is in large measure describes the function of this biological membrane. All of the membranes in the cell, the internal ones and the external ones, have a similar basic structure. Okay. They're made of phospholipids. I apologize for the typo. And they're arranged in a specific configuration, as we'll see. The membranes also contain proteins. There's a lot of proteins that are involved with the membrane and just below it. They maintain structural integrity for the membrane. They provide transport functions, receptor functions so that signals can be received by the cell. They allow cells to tell your immune system that they belong there instead of being destroyed by the immune system. That's what the sugars are doing. All right, so there's a lot of functions in lipid bilayers or membranes of cells. This molecule is a phospholipid. All right, this is the basic molecule that you find in cells. And what's happening here is that you've got a typical lipid. Here's the glycerol left over, and the fatty acid tails. One of them is saturated, the other one's unsaturated. All right? But where a fat would have a third fatty acid tail, this molecule doesn't. Instead, it has this head group, which is highly polar. There's a phosphate on it, which is negatively charged. That's why we call these phospholipids. There's a nitrogen up here that's positively charged. So there's lots of charges in this molecule, and it interacts very readily with water. Okay? So the heads will interact with water, but the tails are still hydrophobic. They exclude water. And so if you're going to get these molecules together, there's a very specific conformation that you would have to have in order to get these things to stay together. Okay? Phospholipids form bilayers. What that means is that the tails here are getting together, and the heads are staying outside where they interact with water. The tails tend to exclude the water from the central portion of the bilayer. Okay? You don't have to do anything special to get these things to form bilayers. You take a whole bunch of phospholipids, you dump them in water, they form bilayers. Spontaneously, they form these lipid vesicles, which look a lot like cell membranes. All right, so this is how they arrange themselves when they're stuck into water. You have these hydrophilic heads, which are interacting with the water on either side. We can think of this top area as outside the cell and the bottom area is inside the cell. And then water tends to be excluded from this hydrophobic internal space. Okay? Water can cross this bilayer fairly easily, but many other things cannot. Ions aren't going to be able to cross it because they're charged, right? And they're going to be excluded by those hydrophobic tails. Basically, anything that's charged, anything that's big and polar is not going to get across very easily. Right? So the heads face water and the tails don't. And that creates a hydrophobic zone. So every lipid bilayer in the cell looks like this. Even the double lipid bilayers, they're just two layers of this kind of stuff going on. Right? There's really nothing different about them. These molecules, lipid molecules in this thing, can move. They can freely move from side to side, and if we're thinking two-dimensionally, out of the screen and behind the screen. Okay, so in 2 dimensions they move very easily. But because of that hydrophilic head, right, they don't flip-flop. Right, you're not going to have a situation very often where one molecule from this lower part of the bilayer flips up to the higher part of the bilayer. The reason is because that hydrophilic head has to pass through a very hydrophobic nonpolar zone. And it takes an added amount of energy to get that to happen. Okay, so it doesn't happen very spontaneously, but it will happen occasionally. Cells actually have enzymes that are called flipases that will flip those phospholipids back to the appropriate layer. So we know it happens. It just doesn't happen very often. If we add in some of the other components of the bilayer, this is the kind of thing that we see. The bilayer is much like a fluid. These lipid molecules, as I said, are free to move around, and they'll do so. They'll diffuse in 2 dimensions throughout that bilayer. Okay? Many of the proteins are able to do that as well. They're like ships floating on the water, and they'll just float around among those lipids. Some others are anchored. See, here's our cytoskeletal elements, and so some of these protein molecules are actually anchored into the bilayer. We have the occasional cholesterol molecule. That's what this is right here in the bilayer. And we have sugar residues on many of these molecules, glycolipids and glycoproteins. Okay? So some proteins are held in place. They don't diffuse very readily. Cells will actually take proteins for a common function and stick them all together in one big clump so that the reactions happen faster. Okay, so we call those rafts. And there are some other components of the bilayer which we'll take a look at. So some other bilayer components include things like transmembrane proteins, proteins that pass completely across the lipid bilayer. There's a number of those kinds of proteins. In fact, those are the most common proteins we find in the bilayer, proteins that pass across. They don't just pass across once. They may pass across many times, 7 times, 12 times. And those proteins that pass across multiple times tend to be transport proteins or receptor proteins. They're transporting materials across. Cholesterol is also present in the lipid bilayer. Okay. The amount of cholesterol that's in a lipid bilayer strongly affects how fluid it is. Cholesterol is real bulky, and so what happens is that if you got lots of cholesterol in there, it tends to bulk up and prevent movement of the lipid molecules. They don't move as freely, and so it changes the fluidity of the bilayer. Okay. It turns out that we can make all the cholesterol we need. Our livers are very good at it, so we don't have to bring in any from our diet, although most of us bring in quite a bit. All right, so cholesterol is one way of affecting how fluid that membrane is. Glycolipids. These are phospholipids with sugar residues attached, and you saw a few of those in the previous slide. Those have a number of important functions. There's a lot of sugar residues on the outside of the bilayer. We never find sugar residues on the cytoplasm side, on the cellular side. Okay, the cell always flips those things over to the other side. The reason is that one of the functions of those sugar residues is as a pH buffer. Okay, sugars have lots of negative charges. So if the pH around the cell changes, those negative charges tend to suck up hydrogen ions. And they move the pH back towards a neutral state by reducing the number of hydrogen ions. Okay, so it serves a protective function. It also serves a recognition function. The sugar residues on the surface of the cell are telling cells of your immune system that that cell belongs there. Immune system cells will recognize it and not attack it. If those sugar residues are not there, or if they're not correct, then your immune system will attack that cell and destroy it. It's one reason why you reject organ transplants. Glycoproteins. You don't just have sugar residues on lipids. You have sugar residues on proteins as well. Some of these may be transmembrane proteins that span the helix, but all the sugar residues are on the outside. And again, they're serving the same kinds of functions as glycolipids are. All right? So, as I said, they're used to identify cells of the immune system, but they also serve as a pH buffer. The proteins in a membrane have a lot of different functions, and these are just a few of them. They may be enzymes, so there may be a specific enzymatic reaction, chemical reaction that has to occur near the surface of the cell, and you would have enzymes that are actually embedded in the bilayer that serve that function. Okay, they're converting substance A to B and B to C and so on in an enzymatic pathway. You may have proteins that are serving as signal receivers. So here's a receptor that receives some sort of signal from outside, and it transmits that signal somehow to a protein that activates a molecule inside the cell so the cell knows the signal's received. Okay, this process we call signal transduction. The signal is being transduced from outside the cell to inside the cell. That's typically how signals are received and processed by cells. We may have molecules that are simply serving as transport proteins. They transport ions across. One of the problems with lipid bilayers is that you cannot get ions across that bilayer. Right? They just won't go across very easily. So in order to be able to bring in things like sodium ions or potassium ions or chloride or whatever, you've got to be able to have a channel, a watery channel through the lipid bilayer. The way you do that is that you put a protein in so that you have an open pore, basically. Okay? We may have cytoskeletal proteins that are close to the lipid bilayer and anchoring some of these other proteins in. The cytoskeletal proteins serve the important function of providing structural integrity for a bilayer. It's a fragile structure. It would break very easily were it not for the cytoskeleton. Right? There may be extracellular proteins that are anchoring into the cytoskeleton across the bilayer. Okay, so lots of different kinds of functions for these proteins in the bilayer of cells. Okay, so what I want to do then is start talking about some of the properties of the bilayer more than just describing what's there. And one of the main things we have to look at now is permeability and diffusion, because these processes are strongly affected by the properties of lipid bilayers. Okay, so diffusion, you're probably already familiar with this, but diffusion is just the tendency for particles to even out. Okay, the air in this room, the air molecules are evenly spaced. There is a small but non-zero probability that all of the air molecules in this room could suddenly sweep over to one side of the room and suffocate half of us. It's a real probability. It's very, very tiny. But the tendency for these particles is to be evenly distributed. That's true in liquids as well. Right? You dump food coloring into a glass of water and you see that the food coloring spreads out until it's evenly distributed in the glass of water. That's diffusion. Okay? So lipid bilayers only allow limited diffusion. Not everything can get across. Right? Hydrophobic molecules, even if they're big, they go across really easily. Cholesterol-based things like steroid hormones. Steroid hormones get across almost immediately, right? Very quick. So hydrophobic molecules can get across. Small polar molecules can get across. Water goes across pretty well. Some cells will actually increase the permeability of water, of the membrane to water. They'll put little channels in the lipid bilayer that let water flow through. Your kidneys do this when you're trying to reclaim water if you're dehydrated. Right? So small polar molecules get across. Ions don't. Large polar molecules don't, for all intents and purposes. Now, in reality, there is a small amount of transport across a bilayer for these ionic molecules, but it's a very small amount. It's certainly too small for the cell to find useful. All right, so in practice, these kinds of molecules simply don't get across a lipid bilayer. All right, so lipid bilayers are selectively permeable. Right? They will allow certain materials across, but not everything across. They're not indiscriminate. And it's just a result of the properties of lipid bilayers. A lipid bilayer, because it's got that big hydrophobic space inside, is not going to let hydrophilic things cross. It takes too much energy to force those molecules across a hydrophobic space. Okay? So the cell is going to have to regulate what's going to be getting across the bilayer. Okay, as far as diffusion goes, as I said, diffusion is the tendency of particles to move until they reach a steady state with an equal concentration. We call that an equilibrium. So in diffusion, as is shown up here in the top part of this figure, these molecules of dye will move across this membrane, if it's permeable to them, until you have an equal concentration on either side. At that point, it's not like they stop. All right? They're still moving across. You're still getting movement of molecules from left to right across the membrane, but you're getting an equal amount from right to left across the membrane. So there's no net change in the concentration. Okay? Molecules will diffuse even if mixed until equilibrium. So we've got these red molecules on one side of the membrane, the green ones on the other side, And you get movement of these molecules across in opposite directions until, once again, you have equilibrium, where there's no net change in the concentrations. So the red molecules are moving from left to right to equalize their concentration. The green molecules are moving from right to left to equalize their concentration. And then at this point, at equilibrium, you've got movement in both directions, but it's an equal amount. So that's why there's no net change in the concentrations. Okay. This whole thing starts to change dramatically if these particles are charged. We won't get into this. In cell biology, we get into that a lot because we talk about neurophysiology. And neurons are able to work simply because they maintain charged states across the bilayer. So when we get these, if these things are charged, then it gets very complicated. Right? But in general, we'll just use the idea that everything moves until equilibrium. Okay? There are some terms that we have to be aware of that relate solute concentrations on either side of a selectively permeable membrane. Okay. Hypertonic refers to higher salt. Hyper meaning higher or greater. Hypo meaning lower. Okay. Hypotonic. If you're hypoglycemic, you have low blood sugar, for example. And then isotonic, which simply means equal. Okay? So a hypertonic situation means there's higher salt, hypotonic is lower salt, isotonic is equal salt. But when we use these terms, we must be extremely careful with them. The reason is because they are always relative. You're talking about two sides of a membrane, okay? Left side, right side. You have to refer to these relative to one side or the other. You can do it both ways. Okay? So these things are relative to the two sides of the bilayer, and I have a few figures to show you how that works. All right? So here is a figure that I put together that shows a membrane of some sort. We've got some dissolved salt on one side and less dissolved salt on the other. Okay? If this membrane were permeable to this material, then this stuff would be diffusing from left to right until you got an equal concentration. That's the way diffusion works. However, this membrane is not permeable to whatever this is. Right? So it can't move. So because side A has more dissolved material than side B, you just count the dots, we'd say that side A is hypertonic. Side A is higher salt, so it's hypertonic with respect to B. But we could just as easily have said that side B is hypotonic with respect to side A, because side B is lower salt than side A. So when you're referring to these terms, you've got to be aware of which direction you're talking. Okay? Are we talking side A versus side B? Are we talking side B versus side A? Right? So it's all relative, and that's why it's so important to specify context when we're talking about solute concentrations across lipid bilayers. Okay? You may think this is straightforward now, but I've lost count of how many people screw this up on exams. They'll reverse it all the time because they're not thinking of relative terms. Okay? I'll say, solution with respect to the cell, or cell with respect to the solution. You've got to be careful. All right, so let's take a look at the situation again. We'll put in some dissolved solute on the left side. Is A hypo or hypertonic to B? It's hypo, right? Side A is hypotonic to B because side A has lower salt than side B. If I had asked this question, is B hypo or hypertonic to A, you would say B is hypertonic. Yes, exactly. Good. Okay? Well, let's change the situation a little bit here. Is A hypo or hypertonic to B? Yeah, it's a trick question. It's neither. It's isotonic. All right? We've got the same number of molecules of dissolved solute on side A as side B. They're the same concentrations. So they're isotonic with respect to each other. They are the same. Okay? Everybody see how that works? Good. Okay. Well, let's add a few more molecules here on side A. Is B hypo or hypertonic to A? Hypo, exactly, because B is lower salt than side A. We would say then that side A is hypertonic with respect to side B. Okay, so we add a few more. Doesn't change anything, right? B is still hypotonic to side A. I'll just increase the concentration on side A. So once again, we'd say that side A is hypertonic with respect to side B. And so that's what I mean when I say it's all relative. You must be very, very careful about which direction you are referring to, which side you are referring to. All right, so we were looking at a membrane that is selectively permeable, and in fact, it wasn't letting our dissolved material across. There's a funny thing that happens when you have a membrane that's not letting solids across. What happens is the water gets across instead. And that's called osmosis. All right, so if you have a barrier that water can get through but salts can't, the water will move to equalize the concentrations of salts. So water then moves from low salt to high salt. Okay? Water moves from low salt to high salt, or from a hypotonic solution to a hypertonic solution. Basically what it's doing is diluting out the high salt. You can demonstrate this, and this has been done many, many times in biology labs. If you have a selectively permeable barrier that lets water through but not salts, right, and you put a low salt solution on the left and a high salt solution on the right, and you let it just sit for a little while, what ends up happening is that the level of or amount of water rises on the high salt side, because water has come from the low salt to the high salt side in an attempt to diffuse and equalize those salt concentrations. You can actually observe that really easily. There's no sort of desire to move. What's happening is that the water, because it's polar, is being attracted by the molecules of salt, which are charged. Okay, so the water is essentially being pulled across by charge interactions. That's really all that's happening. Okay, the pressure generated by osmosis can actually outcompete gravity. You'll see situations where the water level rises on the right side of this tube, fighting gravity, because the water pressure is greater. All right, so that's osmosis. It's the movement of water through a membrane that allows water across but not other materials, or only selectively allows other materials across. Big, big consequences for cells, though, because cells are surrounded by lipid bilayers, which are selectively permeable membranes. They let water across, but they don't let a lot of other things across. A lot of charged stuff is going to need help to get across. So cells have a big problem with this. Okay? If you're in an isotonic state, the cell is in a solution that is the same salt concentration as itself, you get an equal amount of water coming in as going out. Right? You're at equilibrium. So the equilibrium state is that there's no net flow of water. Equal amount in as goes out. For a plant cell, that's not a good situation, because the plant cell is going to wilt a little. It's not going to be able to maintain pressure against the cell wall so that it stays upright. If you take a cell and put it into a solution that is hypotonic to it, right, low salt in the solution, high salt in the cell, then what happens is the water moves from low salt to high salt. And since the high salt is inside the cell, the water moves inside the cell. So you get more water going in than coming out, and the cell, if you're dealing with an animal cell, the cell is likely to burst. If you're dealing with a plant cell, it's a little bit different situation, because a plant cell has this rigid cell wall. So what happens is water comes into the cell, builds up water pressure, pushes against the cell wall, right? And that keeps plants from wilting, that water pressure. Right, so we call that a turgid state for a plant cell. On the third situation here, if we put the cell into a solution that's saltier than it is, right, the cell is hypotonic to the solution, or the solution is hypertonic to the cell, then what happens is the water goes out. Water moves from low salt to high salt, and in this case, the high salt is outside the cell. So the water moves outside the cell loses water, shrivels up. In the case of these red blood cells like they're showing here, what happens is the cells get spiky because the cytoskeleton can't collapse. And as the water is lost, the cytoskeleton is pushing up against the lipid bilayer. For a plant cell, this is an equally disastrous situation. It shrivels. Okay, the plant wilts. Notice that the plasma membrane, the cell membrane, is attached at certain points to the cell wall. So it doesn't completely pull away, right? But it does shrivel. So you get this open air spaces between the membrane and the wall. Okay, so if the cell is hypertonic to the solution, it gains water. If it's hypotonic to the solution, it loses water. What that means for an animal cell, for example, is it's got to maintain an isotonic state or it's in big trouble. All right, so how do cells deal with this? Plant cells, no big deal. All right, they have a rigid cell wall. It's unlikely that they're going to burst, given that that cell wall is a very strong, durable structure. So plant cells can absorb quite a bit of water. They expand. That water exerts a pressure against the cell wall, and as a result, the cell wall is very rigid. Okay? Animal cells typically are bathed in isotonic fluid. Right? Our cells, the salt concentrations in our cells, red blood cells, for example, are typically isotonic to our blood. Right? So that way they don't burst. Some other cells in our bodies, however, do not maintain isotonic states. In fact, they cannot if their function is to be maintained. Nerve cells, neurons, cannot maintain an isotonic state. If they did, they would not function. So they have to keep from maintaining that state. Single-celled organisms constantly have to worry about getting rid of water if they're fresh water, or taking in water if they're in the salt water. Okay? The reason for this is that if you have a single-celled organism that's living in fresh water, single-celled protist, it is saltier than the water around it, and so it constantly is taking in water. It's got to get rid of it or it bursts. For a saltwater single-celled organism, the problem there is that the water is saltier than it is. Right? So it loses water, and it's got to constantly bring water in order to equalize the amount inside and out. So these free-living single-celled organisms have a devil of a time having to try and deal with these issues. Okay? Most cells can also pump ions. They can move sodium, potassium, chloride ions across the bilayer so they can equalize concentrations of ions instead of having to try and deal with water. Right? That's one way to do it. If you can't deal with the water flowing across, then you do the next best thing. You move ions across until the ion concentrations are equal. And at that point, you're okay. All right, so cells will use transport proteins. Transport proteins are just proteins that form a channel across the bilayer. Right? Since a bilayer is impermeable to most salts, what you have to do is you have to make a channel, a watery channel through which a charged molecule can pass. Right? That's what transport proteins do. Okay, this is an example of what we call facilitated diffusion. All you're doing is basically making the membrane permeable to the salt. Once that happens, it'll flow across until you get equilibrium. At that point, you have as much flow backwards as you do forwards, so there's no net change. Okay, so that's why we call it facilitated. You're facilitating it by putting in a protein channel, but it's still a diffusion process. It's really all it is. Okay, no energy is expended, but you can't go against a gradient. Right, remember gradients. Gradients are situations where you have a concentration difference across a barrier. A dam, the dam that I showed on Friday is a good example. You've got lots of water on one side, less water on the other side. It's a gradient. Okay, because the dam is impermeable to the water. So this process cannot go against a gradient. It just allows flow until the concentrations are equal. At that point, there's no net change. Okay, so if you want to fight a gradient, if you want to actively transport ions outside of a cell or into a cell against a concentration gradient, you have to use active transport. Active transport is similar to facilitated diffusion. They both use proteins. That's where the resemblance pretty much ends, because active transport is using energy to move materials across the bilayer. Right? The basic process is that the solute inside the cell binds to this transport protein. ATP will transfer energy to the protein so that it can pump that ion across the bilayer. Right? Remember, when you break a phosphate off of ATP and transfer it to another molecule, you're transferring the energy in that unstable bond. Okay? So the phosphate energy is transferred to this protein. That allows it to pump the ion across the bilayer against its gradient. Because the phosphate is still there, we can bind perhaps a second solute that's outside the cell, break the phosphate off, therefore releasing the energy in that phosphate bond, and as a result, we simply pop the protein back to its original conformation, and that solute is released into the cell against its gradient. Okay, we often call these things ping-pong proteins because they seem to sort of ping and pong between two different states. State facing outside the cell and the state facing inside the cell. Okay, but the main difference here is that this thing is using a gradient, using energy and fighting gradients. Right, so, for example, if there's a lot of sodium outside the cell could still move more sodium out of the cell against the sodium gradient because it's using energy to do so. Okay, in this figure, we're showing transport in two different directions, obviously. We call that antiporting. But the point of this is that you can use ATP, go against a gradient, and make the process irreversible. So if this protein happened to ping to the outside state, it wouldn't immediately pong back and put that solute right back inside the cell. By using ATP, you force the molecule to go outside the cell, and you force the outside solutes to come in. Okay? ATP makes the process irreversible. It would take too much energy to go backwards. Okay. So how are facilitated fusion and active transport similar? The answer here is B. That's about the only similarity between facilitated diffusion and active transport. They're both using proteins to do the transporting. Facilitated diffusion isn't really using energy because all it's doing is allowing a gradient to equalize. Okay? Active transport can fight a gradient, and therefore it has to use ATP to do so. So the first statement up there, they both expend energy. That's not correct. Both moving solids against a gradient. Again, not correct for facilitated diffusion. It only allows equalization of a gradient. You get equal concentrations on either side of the membrane. They don't both require ATP. Facilitated diffusion channels are passive. When they're open, stuff goes across. Okay, and then of course E, saying neither one can generate an equilibrium of solid, is incorrect, because that's exactly what facilitated diffusion does. Okay, there are some ways that cells can move materials, large amounts of material outside and inside the cell. Exocytosis is a process of moving materials out of the cell. Fairly simple process. All you're doing is taking a lipid vesicle that's full of stuff, you fuse it with a lipid bilayer, and it opens up, and you get all your stuff going outside. Okay, the opposite process is this, endocytosis, where you bring materials inside. It's essentially the reverse process of exocytosis. Here the lipid vesicle is pinching off, and then it's going to be delivered to wherever its final destination is, usually a lysosome in this case. Okay. How do cells know where all this stuff is supposed to go? That's been a mystery for a long time, but in the last 10 years or so we've started to get a handle on it. There are site-specific proteins that are put into that bilayer that basically dock with the appropriate target. Right? So that's how the cell is able to direct all of this material to the correct locations. Right, so there's three types of endocytosis that cells can do when they're bringing material in. Phagocytosis I talked about in the last chapter, chapter 4. That's where you get uptake of large material. Amoebas do this, for example. There's some food being ingested by an amoeba. Pinocytosis usually brings in small vesicles pretty indiscriminately in terms of the material. Whatever's outside just gets brought inside. So here's a whole row of what we'd call pinocytic vesicles that are bringing stuff in. All right, so small materials are coming in. The other thing that cells can do is receptor-mediated endocytosis, which is happening here. Okay. You remember I said that there are some proteins in the cell membrane that are receptors, and they bind to specific molecules. All right, so that's what's happening with receptor-mediated endocytosis. You have a receptor that binds to a certain molecule. Okay, so it binds to a specific molecule, and then once those molecules have been bound, the receptors are just clustered, and they're brought into the cell by endocytosis. Okay, so for an example, our LDL receptors work this way. LDL, low-density lipoprotein, this is the so-called bad cholesterol. Well, they're all bad, but this is worse than others. This is the stuff that gives you plaques in your arteries, clogs them. There's our LDL particle. It's jam-packed full of cholesterol. Nasty stuff. It's insoluble. That's why it forms plaques in your blood if it gets out. So to protect the circulatory system from this stuff, it's surrounded by a phospholipid outer layer. Right? That makes it interact well with the watery blood that it's flowing through. There are proteins on the surface of this LDL particle that are going to target it for receptors on the cell surface. So when this thing bumps into a cell plasma membrane, it binds to those receptors. What happens then is the cell receives or perceives that those receptors have been bound, and it forms a little vesicle. Okay? And that vesicle then pulls the cholesterol into the cell. It goes into lysosomes, and then it gets targeted for various cell membranes in the cell. Right? So receptor-mediated endocytosis is a very common way of bringing particular materials into the cell. Other indiscriminate methods just use pinocytosis. Right? So Cells have these receptors to bring that material in. If you don't have the receptors, or you don't have the right proteins on the surface of the LDL product, you have familial hypercholesterolemia. I've known people in their teens that had cholesterol levels of 300 milligrams per deciliter or so. It's ugly, right? You want to take Lipitor at that point. It's deadly. Okay, so we will start Chapter 6 on Wednesday.